All right, let me stand out of the way. Okay, so rather than me trying to sync up typing something on a piece of paper while we go to the other one, I think one of the issues with that kind of lesson or instruction on how to do something is that the information doesn't stay on the board. Some of you write really fast and you're copying it, and that really, using your hands and copying it is actually when you remember it longer. For some of you, you're screenshotting it. Screenshots help, and I do use that sometimes in some of my training, but the truth is I haven't learned it. It's on a JPEG someplace else, and then I have to find some more time to go and take a look at it. So I actually get less out of screenshotting when it's instructions than I get from actually trying to jot down the parts that I can. For that. And so the other part is, part of the reason that big classrooms work with the huge whiteboards is that if you're not using it all on PowerPoint, turning down the lights and then having it go slide to slide, then everything that you have already written is already still there. And I'm going to try to keep this on this board as well as I can and have this get as close as I can to just a little bit further down in here to keep it as focused as possible. Okay, and me out of the way. All right, so I did mention on the very first day of this that one of the challenges for this particular unit was chapter 5. Not because chapter 5 had a whole bunch more vocabulary words, and not because chapter 5 wasn't completely doable for students, but because most students, and that doesn't mean you, and it doesn't always mean that it isn't an okay way to go, but because most students do not complete a lot of activities, especially reading and reviewing, until it's very, very close to the exam. And when it's all new information and you're doing your reviewing or you're putting together some notes to jog your memory and it's very close to the exam, you just don't hold as well. You just end up getting a little bit more confused. So one of the things that I like to do with face-to-face, and right now we're basically face-to-face, is give you some really targeted information about what, in best case scenario, your outline or targeted kind of notes should look like. So that's really what this is like. Kind of like the video lessons that I did when I had a giant whiteboard for putting together some notes for chapter 3. Okay, so chapter 5 is got a general title, which is kind of long, Forces of Evolution and the Formation of Species. So in other words, it really is about how evolution works. What are the main things that we've looked at, most of them, but not all of them, and how does that result in variability within a population, but then only some of the variants pass to the future and others of them don't pass at all, or some of them don't pass quite as commonly as others. So how does that all come together when we define a species? Because again, in order to investigate how we're related to other members of our order Primates, or how our ancestors have evolved, right, change in the frequency of alleles over generations to become us, we just need to take a look at a little bit of this. So these 3 terms over here to the side in purple, and especially the big star here, population. The unit of evolution is the population. It's not the individual. You are not evolving. When the 2 gametes met and fused, your code was set. So the change for the species doesn't happen until your 50% contribution, right? Principle of segregation and principle of independent assortment plus crossing over, until that passes to an offspring, which is why fitness in this particular conversation really is bottom line, who reproduced, were they able to get a grandchild generation out of it, and did they reproduce more successfully or more times than certain other individuals? So population. Chapter 3, unit that matters the most of genetic code is the base pair sequence. That's the little tiny bits of code. Chapter 4, when we're looking at inheritance or heredity, the unit of inheritance of heredity, that in the way that we study it, is the gene and its allele. And then chapter 5, the big concept here for 5 is this is all based on population. When we finish up with this and we come back, just save this term, we're going to learn what a DEEM is. And this is what we look at statistically when we are looking at how a population evolves over time. And then we're going to finally give you a usable definition of what a species is. We're going to use actually technically one that's very specific called the biological species concept. There are other ways of defining a species. We're using this one for this point in our course because right now we're not looking at extinction or other kinds of things here. Okay, so just hold on to those. All right, firstly, as the chapter starts up, let me grab image script so I can refer to some pages to you. 1, you're going to go from the beginning of chapter 5 through page 99. After 99, I'm not going to ask you any questions. So if you're taking lab class, yes, lab class works on the last little bit of 99 in regards to the mathematical formulas, but we don't have time for do that, and this isn't a lab class, and it doesn't have a math precondition for you to enroll, so we're going to leave that one for lab. This one, again, the sections starts on 83, and you're going to go through 99 and stop. Okay, so we've been talking about variation a lot so far. So first thing in your notes, I would say letter them or number them any way you want. But our first big concept here, try to write it boldly, for chapter 5 is variation. That would be your big term here, variation. Where does genetic variation come from? It's the first part of it. We're looking at page 84 on this part of the conversation. So we've got 3 things going on. Or maybe, let's say we have 4 things with some subsets going on in this particular section. My suggestion is for this one, this be a separate outline and put those notes separate from the whole rest of the chapter. When you do it that way, if you spend a few minutes reviewing and you look these over for a few minutes, then you're not getting that internal snow globe of remembering everything that was in the chapter, but just one conversation that was in the chapter. And this one has been additive as we go. So our big subcategory here is, what is variation? Variation is mutations. Right, that's the source of it. We don't get anything new if you don't have a mutation. And a mutation is the only source of a brand new gene. So now you've got 2 more little things that we've already done. Just want to make sure that you know that they've already been in here. So sources. And types. Of mutations. So sources of mutations and types of mutations. This chapters 3 and 4. You're going to have this already from some other things that you've looked at. Our types of mutations, I'm not going to get this too crowded for you right now, but we just did them in the first hour and we talked about them in the last class meeting. Our types are point mutation, chromosomal structural mutation, and chromosomal numeral or abnormal chromosome number. Those would be our types. I'll just put 3 here then to jog your memory that you have that. Sources. Sources here. If we're talking about evolution, right, how evolution works, then it's just mutations that occur in gametes. So the source are going to be a couple things that happen during meiosis, right, which is gamete formation. Mendel took a look at a couple of these. So Mendel identified some of these changes or outcomes of the changes. But for us, we've got, right, the changes that we looked at here. And then I mentioned also earlier, which you probably already have in your notes, we have crossing over. That's also in the meiosis video. You all, I would definitely watch that meiosis video if you haven't already, the one that's in Canvas. Perfect. It's got lots of potential questions that you're going to see on Wednesday. So meiosis, crossing over, every single chromosome you have has small point mutations because when the pairs line up, they swap. And it's every single chromosome and it's in multiple spots. So there's hundreds of them every time you make a gamete. So that's one of them. And then the other source would be non-disjunction, when we get some things that are clinging or not working correctly, where we've got breakage, or we've got unlinking linked genes and that sort of thing. So these are the ones that sort of happen. And then we've also got just general, everyday, small point mutations that make small changes that don't necessarily make huge variables. Now, one last thing I did in order to emphasize the idea of mutations, we did look at things that make the function of body parts harder for the individuals that carry those. But remember, if it isn't a giant change, if it's just point mutations, it might be neutral. That's probably statistically the most outcome, or it can be beneficial. We looked at the ones where it broke, something moved, something's missing, or something's added, and those are not beneficial. Those are going to be deleterious mutations. Okay, so that's that first part of it. Now, to add two more things that are going on early on in the chapter that you want to have related to this, variation and mutations. We're going to drop it down from mutations. We have variation that does not end up working within natural selection. So what we can put is below this too. I'm going to just do this one as an A, non-selective. And then B will be selection, right? Remember, natural selection. So for non-selection, these are going to be changes or variants, right? Coming back to this one, variants that do not create a new species. They don't necessarily benefit. So the population, they're just variation. Remember, variation is any change in a gene or an allele. Any change in a gene or an allele. So for non-selective ones, I'm going to drop it down here. And real quickly, be sure you have some notes on gene flow. Drift. Genetic drift. And in your reading, there are two forms of this. Gene flow, drift, and sexual selection. And then, of course, mutation. Mutation is random change. Variability. These are the aspects of variability that's in the entire inventory of a species. It's all the types of variants, but they aren't the part of the process that's the test. This is just variation. When these exist, is the species evolving? Yes, because evolution doesn't have a direction. So evolution is just change in the frequency of a gene or allele over time. So these are a form of evolution, but they aren't the form of evolution that helps a population to become better adapted. Now, the next part, selective, think of it as a test. Get down here. So selection, right? So we have all the variants are present. This is always happening all the time. And then nature, your current natural environment, goes through selection. There, and selection is here. So you can think of it as natural selection. You can write it natural selection or just selection here. Selection is going to, I'm going to do this a little bit. Yeah, you can see the orange one. A little bit brighter here. Say natural selection in a different color. Natural selection does not work on the phenotype. It works on the genotype. It works on your genotype? No. Phenotype. The natural environment doesn't know all of your recessive genes. You don't know all of your recessive genes. So the natural environment just selects from the output. What is the result? What is the result of what you have? So genotype, nope, doesn't work on the genotype. Works on this one. And one more item here for this first outline. Natural selection. Natural selection has actually two forms of it. So we've also got two more things here. Down this way. It's in your reading. Okay, so the two forms are stabilizing and directional. All right, natural selection. It's not always looking for something that's different. So stabilizing, and I didn't spell it right. Oh, that's the beauty of the whiteboard, isn't it? And directional. All right, so natural selection is the test from the pool of variation. That's why I did non-selective first. So you accumulate variation. Variations come from mutation, and then other things happen to shuffle or reorganize those variants. Gene flow, drift, sexual selection, and mutation. And then when this is part of the phenotypes of a population, then nature selects. Take this out now so it's a little less busy. Natural selection. Works on the phenotype. You have stabilizing and directional. If you remember from chapter 2, which in summer school, that is one of the better things. It goes really fast, but as you are remembering every single moment that we're in class, that part's good. You don't forget things, but it also makes a rush for your day-to-day schedule here. So natural selection just selects on reproductive success. So it is a fit to the current natural environment. Well, if your species population has been in an area where the current natural environment hasn't changed very much for 10,000 years, then every new inventory of offspring, trees, plants, bugs, whatever it is, goes to the same test. So that's when it's stabilizing selection. If the environment is changing, or you have a series of, just a century where the environment is a lot colder, then all of a sudden a different selection of phenotypes are the ones that are most favored. And there you get directional selection. That's why evolution doesn't have any common direction, because the environment fluctuates. Okay, that would be your first suggestion for making some outline notes on this variation, the topic of variation. And it fits all the things that we've done before, and then also adds that you want to look for these three that we haven't talked to. But the only source of a new gene is mutation. That's the only place where something new comes from. What's the outcome of gene flow? Gene flow actually moves alleles from population to population, and it keeps them more similar over time. Genetic drift is random. So drift is the opposite of natural selection. It means that a gene or a phenotype is more or less or stable or just appearing, but it doesn't have anything to do with improving the benefit of the individual for surviving and reproducing. And then sexual selection is when some species, like humans, pick for characteristics that aren't necessarily helpful for that individual to be healthy and have good longevity, but we want to choose for those things anyway. Okay, all right, so I'm going to erase this part and go to the next page that I think would be a very helpful way to outline your reading. Okay, so the first one was, I'll leave that first concept here. Now the next one that you'll work on, that's the benefit of those classrooms with the giant boards, right? We can leave it all up for the whole time. But these next little parts will be briefer, and then we'll get to whatever part of the lesson I can do for some time limits here. Now, the second section, let me check again. I do believe it starts on page 94 through 96. So that first one was long because that goes up to, yeah. Now, so you're going to start again on species concepts, 94 to 96. In the section in between classification and evolution, I already had you prep and answer questions about the taxonomic chart. So you're not going to see any more of those questions like what family do you belong to, or what is your tribe for right now. So we're going to start using the taxonomic chart at the end of this week when we get into chapter 3. But for right now, we're not going to rememorize that. You're not going to have that at your fingertips. I'm going to stick on this particular conversation right now. So next big question for a couple pages is what is a species? So 94 to 96. There are a variety of ways of defining a species, and we'll come back to the details of this when we finish up our last little bit of time for lessons. But for us right now, bullets on this one, and I would, just for ease of use and quick reference, make this its own separate sheet. We are using the biological species concept. Biological species concept. And then you'll have just the definition for that. We'll do that. Then there is a bit of a question then from this one. If we know what a species is, then the next big question that we have is, how is a new species formed? How is a new species formed? Real quickly, and then you can fill in the rest of it when you do this. And make sure that you have in your notes that first it has to stop gene flow, add mutations, and then the two of these have to create rims. Reproductive isolating mechanisms. They're a quick list in your reading. It's at the bottom of 96, table 5.2. So you're looking for table 5.2. Be sure that you recognize this acronym and you know it means reproductive isolating mechanisms. In other words, why can't two similar species mate? Even if they look very similar to us, let's say we're looking at two different species of small brown monkeys that live in trees and have a long tail, why is it when you go to the zoo and you look at them, they have two different species designations? That's because they have developed in different populations a rim. So you've got pre-mating isolation. There'll be 4 of them there. Just once you look at them, they'll make total sense. And then there's post-mating. Those are 4 basic ones we'll look at, and they'll be right there too. Okay, that would be your second structural outline for that one. Okay, so 2 more I would do, and the last 2 are going to be, you can put them together in one section of note taking. So our last part of the chapter. What is the shape of evolution? Frequently you'll see some of these charts or mode, I'll say mode in these 2. For these, this is going to be on 97. And as you look, it has the 2 little diagrams there. Please make sure that you look at the diagrams. The figure is 510. For this one, it goes through that with a little bit of vocabulary. So if you see something where there's a prefix and the second part of the term, the root term is genesis, right? Genesis means to begin something, from where something comes from. So there's that part. And the last one, what is the pace of evolution? I want you to just look at 2 paces of evolution, if that makes any sense at all as we're looking at these. Just 2 ways that we can look at how this works. Make sure that I'm not forgetting anything else that I wanted you to take a look at. No, I think I give myself little notes so that I put together as I write up some questions for you for things. So in PACE, it'll be page 98, and it says the tempo of speciation. And I just did see something else. So we've got gradualism. Who put that? Let's make sure I'm writing it more clearly. Who put this idea together, and what is it they think is happening? And the other one is punctuated equilibrium. And you see it punctuated, equilibrium. I've been out of practice from writing on the whiteboard. My handwriting is getting sucky. Okay, gradualism and punctuated equilibrium. So this would be your big third one. There's not as many vocabulary words, but there are some diagrams and illustrations there for how that might actually come together. All right, so when I said that I looked at something, and I wanted, okay. One more thing. I'm going to use for this. Okay, so this is how I would put together my notes. I would make sure that the third one has shape and pace and those diagrams. The second one, definition of the species. How does it form? Gene flow, mutations, and RIMS. That would be your second one. And then the longer one that we put together for variation. Okay, hopefully that really, really helps. And as you know, I've already, of course, recognized that in the type of format that we're working on now for exams, very targeted, clean, not messy, not long sentences, but putting together these targeted outlines where you have just a couple of little ideas that can jog your memory on the type of exams we have, they really help. If they're too long, they're not going to help. If you are writing stuff out and you don't have a chance to practice it, or you've got too many conversations all at the same time, it also doesn't help in a time test. And then the last one, looking things up once you start the test, that doesn't really help. So making your own really good outlines, notes, or things like that that you can refer to, that is what, right? And if you've made it, then you've learned it, you can use it, and I am all fine with that. I think that's a normal way that we would go through this particular process. But again, once you log in on Wednesday, you can log in within the window. You'll have about 45 minutes to complete the exam, and most of the questions are going to be really, really brief, like how many genes have we identified in humans so far? To about 20 to 25,000 genes. How many chromosomes do you have in a diploid cell? 46 chromosomes, right? Those are the 5-second questions. Okay, all right, I'm going to stop this.